This video tutorial is all about cloning, specifically artificial clones in animals. Successful cloning starts with cells that are totipotent. Now, totipotent cells can divide and differentiate into all types of cells found in the adult organism. So in animals, the only truly totipotent cells are found in very, very early embryos. Reproductive cloning can produce large numbers of genetically identical animals. Now, this can be really useful. So, for example, if you have an elite farm animal produced through selective breeding, you might then want to clone that to provide multiple copies, a whole new generation of calves that share exactly those traits that you have selectively bred to get even more elite farm animals. Or in the case of the spider silk making goat, a genetically modified organism, one that has this very unusual characteristic. Once you have acquired that characteristic, you could then clone that spider silk making goat so that you can have more goats that are able to do that same skill. Now, mammals can produce identical offsprings, i.e. twins, if an embryo splits very early in development. This process and our understanding of it has given rise to an artificial cloning technique that has been used since the 1970s. The first thing that happens is you take a zygote, so a fertilized egg, and you create that in vitro, so through in vitro fertilization. The zygote is allowed to divide by mitosis to form a small ball of cells. And then those cells, which are still totipotent, are separated out and allowed to then continue dividing separately. Each small mass of cells is then placed into the uterus of a surrogate mother. This technique has been used to clone elite farm animals or animals for scientific research. However, the precise genome and phenotype of the offspring produced will depend upon the sperm and the egg that are used. So the actual precise phenotype of the offspring here will not be known until the organisms are actually born. Somatic cell nuclear transfer is another artificial cloning technique. Now, it's the only way we currently have to clone an adult. The advantage is that the phenotype is known before cloning starts. And this process was first performed successfully on a mammal in 1996 to produce Dolly the sheep, which hopefully you will have heard of before. So what actually happens? Well, an egg cell is obtained and its nucleus is removed. And this is known as enucleation. Then a normal body cell, so a somatic cell from the adult that you're wanting to clone, is isolated and may have the nucleus removed. The complete adult somatic cell, or the nucleus that has just been removed, is fused with the empty egg cell, so the enucleated cell, by applying an electric shock. The shock also triggers the egg cell to start dividing as though it has just been fertilized. The egg cell will then undergo mitosis to produce a small ball of cells, and the young embryo will then be placed into the uterus of a surrogate mother. Non-reproductive cloning is the production of cloned cells and tissues for purposes other than reproduction. One example of this is therapeutic cloning. New tissues and organs can be grown as replacement parts for people who are not well. So, for example, skin can be grown in vitro to act as a skin graft over burnt areas. Clone cells have been used to repair damage to the spinal cord of a mouse and to restore the capability to produce insulin in the pancreas. And there's also the potential to grow whole new organs to replace diseased ones. Tissues grown from the patient's own cells will be genetically identical and so avoid rejection, which is the problem when transplanting donor organs. Now, cloned genetically identical embryos can be used for scientific research into the action of genes that control development and differentiation. They can also be used to grow specific tissues or organs for use in tests on the effect of medical drugs. However, the whole of this process is highly controversial, and there are several arguments both for and against artificial cloning in animals. Just to run through a few of the arguments for and then against. So, positives. You can produce a whole herd of animals with a high yield or showing an unusual combination of characteristics, such as the goat mentioned earlier, or the bulls, the sort of stud bulls. You can produce genetically identical copies of a very high value individual and retain the same characteristics. Using genetically identical embryos and tissues for scientific research allows the effects of genes and hormones to be assessed with no interference from different genotypes. Testing medical drugs on cloned cells and tissues avoids having to use full animals or testing on humans. You can produce cells and tissues that are genetically identical to the donor, which can then be used for repairing damage caused by disease or accidents. And individuals from an endangered species can be cloned to increase their numbers. However, the lack of genetic diversity and the lack of genetic variation may expose the herd to certain diseases or pests. Animals may be produced with little regard for their welfare, which may have undesirable side effects such as meat producing chickens that can't walk. The success rate of adult cell cloning is very poor, and the method is a lot more expensive than conventional breeding. And cloned animals may be less healthy and have shorter lifespans. There are ethical considerations regarding how long the embryo survives and whether it is right to create life and simply then just destroy it. And it doesn't promote genetic diversity.